I'm excited to walk you through how we can model the vascular environment in vitro, specifically how shear flow microfluidic systems help us study hemodynamics more accurately than static assays. By the end of this session, you'll learn how to build more physiologically relevant vascular models using shear flow. You'll also learn how to apply practical best practices for shear flow experiments in terms of setup, shear rate selection, pulsatile flow, and real-time imaging. And we'll review real-world data showing how mimicking physiological conditions enhances our understanding of the vascular environment to support the development of more effective therapeutics. So let's get started. Blood homeostasis is essential for survival. Under normal conditions, when there is a damage to a blood vessel, platelets detect a drop in pressure and begin secreting clotting factors that create a platelet plug that ultimately becomes a stable clot. When this process becomes dysregulated, thrombosis can occur, blocking vessels, cutting off blood flow to vital organs like the heart and brain, and potentially leading to death. In fact, thrombo-occlusive vascular diseases are among the leading causes of death in the US and the world. While antithrombotic drugs are essential, every current option carries the risk of bleeding. This underscores the need for new therapies that prevent thrombosis while preserving healthy hemostasis. Yet, 90% of drug candidates fail during clinical trials due to poor translation between preclinical models and human biology. And specifically, in antithrombotic drug discovery, many compounds never progress beyond the discovery stage because we lack organized, reproducible assays that replicate the dynamic vascular environment. And this is because traditional in vitro tools and models have many limitations. For example, murine models differ from humans in key platelet receptors. Also, point-of-care assays such as stick tests and aggregometers provide quick readouts but lack the complexity of blood endothelium interactions and the influence of shear flow. And more physiological flow studies typically require complicated pump and tubing setups that are impractical. To close the translational gap, we need platforms that incorporate shear flow and better mimic the vascular bed. The BioFlux system addresses these needs by providing controlled laminar and pulsatile shear flow, built-in temperature and gas regulation, and real-time live cell imaging, all in a simple, low-maintenance microfluidic format that supports the growth of cell monolayers and the addition of whole blood or other blood components. Together, these features enable more precise, predictive, clinically meaningful vascular models. So how does the BioFlux work? The BioFlux controller is an electropneumatic benchtop pump that generates controlled air pressure, regulates temperature, and drives shear flow through microfluidic channels within BioFlux plates. These experiments combine controlled shear flow with live imaging to capture hemodynamic events in real time within BioFlux plates, which are SBS standard well cassettes containing microfluidic flow cells bonded to a thin glass cover slip for high-resolution imaging. The microfluidics in the plates vary in their architecture and purpose and can support anywhere between 3 to 24 experiments per plate. During an experiment, reagents, cells, whole blood, or other blood components are loaded into the plate wells. The plate is then mounted onto an inverted microscope and connected to the plate interface. Through the interface, controlled air pressure generated by the BioFlux controller drives small fluid volumes through the microfluidic channels at defined shear rates. Flow can be either laminar or pulsatile, generating pulses that can cycle to up to 2 Hz. Throughout the run, automated time-lapse imaging captures dynamic cellular responses in real time. We've reviewed how the system works. Let's look at published studies that have leveraged the BioFlux to model vascular processes and hemodynamics. In the first study, we can see how the BioFlux's ability to deliver controlled shear flow made it possible to examine a pressure-sensing ion channel in platelets called piezo-1. The goal was to understand how piezo-1 contributes to shear-induced platelet activation and arterial thrombosis. To explore this, stained platelets from a piezo-1 knockout mouse model were flowed over collagen-coated microfluidic channels in 48 well bioflux plates at 20 and 60 dynes per centimeter square, which mimic arterial shear rates. As we can see in panel A, platelet aggregation dropped significantly in the knockout model as compared to the wild type. To then confirm this mechanism in human platelets, human platelet-rich plasma was flowed over collagen-coated microfluidic channels at the same shear rates as before, but this time in the presence of a piezo-1 agonist, YOTA-1, and various doses of PNS, an anticoagulant. Panels B and C show that YOTA-1 activates human platelets by increasing calcium influx, shown in green. And importantly, PNS reverses this activation, confirming the involvement of piezo-1 in platelet activation and thrombus formation. This study highlights a key point. Understanding how a pressure-sensing ion channel regulates platelet activation simply wouldn't have been possible in a static assay. The bioflux enabled precise, physiologically relevant shear flow conditions that were essential to discovering this mechanism. Second study, the bioflux was used to model endothelial injury under shear flow to investigate heparin-induced thrombocytopenia, or HIT, which is an autoimmune disorder marked by a dangerous hypercoagulable state. The researchers wanted to test whether modifying the HIT-like monoclonal antibody, KKO, specifically by deglycosylating it, could reduce thrombosis by preventing KKO from activating platelets through its binding to PF4. To do this, they grew a monolayer of human umbilical endothelial cells in the microfluidic channels of 48 well bioflux plates. Once the monolayer was established, they induced endothelial injury using a blue light photochemical approach while perfusing hematoporphyrin. After the injury, whole blood treated with the HIT-like antibody KKO, as well as other HIT antibodies, was flowed at 10 dynes per centimeter square over a period of 15 minutes. The results showed that deglycosylated KKO prevented thrombosis when the blood was treated with the HIT-like antibody KKO, likely competing for the PF4 binding site. Similarly, DGKKO treatment prevented thrombus formation when other HIT antibodies like 5B9 as well as HIT 1, 2, and 3 antibodies were tested. This work demonstrates how the bioflux allows researchers to incorporate endothelial contributions to hemodynamic processes under shear flow, something that's hard to achieve with traditional in vitro tools. Next, we show how the bioflux was used to investigate neutrophil extracellular traps, or NETs, and their contribution to thrombosis in HIT. Neutrophils activated with TNF-alpha were flowed over fibronectin-coated microfluidic channels in 48 well bioflux plates. The neutrophils were then incubated overnight with PMA to induce NET formation. Afterward, the channels were perfused with PF4 and DNase-1 at a shear of 2 to 5 dynes per centimeter square. Panel A shows that NETs became more compact when PF4 was present. In the absence of PF4, NETs look more diffuse, but when PF4 is present, NETs became more compact and their tails became more prevalent. This change in structure of NET PF4 complexes led researchers to hypothesize that NETs would become more resistant to DNase digestion. Panels B and C confirmed this hypothesis, and when DNase is applied in the absence of PF4, NETs were eliminated, and the opposite happened when NETs were treated with PF4. This resistance to NET degradation might underlie the hypercoagulable state present in HIT. This is a great example of how the bioflux supports modeling diverse hemodynamic processes, including NET formation, which is increasingly recognized as a key player in inflammatory and thrombotic disease. In the next study, the bioflux was used to evaluate a nanomedicine system designed to deliver plasmin directly to fibrin-rich thrombi. This targeted approach could reduce the bleeding risk commonly associated with antithrombotic therapies that have to be delivered systemically. To evaluate the nanomedicine delivery system, plasmid-loaded nanoparticles were treated with thrombin. They were flowed over preformed thrombi on collagen or von Willebrand factor-coated microfluidic channels for 30 minutes at 25 dynes per centimeter square. After the flow experiment was over, outlet well samples were collected to further quantify fibrin degradation using a D-dimer ELISA. As we can see from panel B, plasmid-loaded nanoparticles treated with thrombin successfully anchored to fibrin-rich thrombi and promoted clot degradation, similar to levels observed when free plasmin was perfused through the microfluidic channels. This study demonstrates how the bioflux provides a controlled environment to test site-directed antithrombotic therapies, helping bridge the gap between concept and clinical translation. In the final study, researchers used the bioflux to compare two p-selecting targeting therapies, crizanlizumab and inclacumab, for preventing vaso-occlusive episodes in sickle cell disease. Whole blood from sickle cell disease patients and healthy controls were flowed over p-selectin-coated microfluidic channels in 48 well bioflux plates at 1 dyne per centimeter square, using a 1.67 Hz pulsatile flow frequency to mimic venous flow during these vaso-occlusive events. Although inclacumab had higher binding affinity to p-selectin, both drugs performed similarly in reducing leukocyte and erythrocyte adhesion, suggesting the safe use of inclacumab as an alternative therapy, especially after crizanlizumab lost marketing clearance in some countries. A key advantage in this study is that the bioflux plate formats allowed researchers to test 9 different drug concentrations, making dose-response studies fast, consistent, and highly reproducible. As we can see from these studies, thrombo-occlusive vascular disease remains a major global health burden, reinforcing the need for better therapies. To develop antithrombotic drugs that are both effective and safe, we need in vitro models that reflect physiology more accurately. Shear flow platforms like the bioflux help bridge the translational gap by offering controlled laminar and pulsatile flow, regulated temperature and gas control, and real-time imaging in plate formats that support up to 24 microfluidic flow cells that can run simultaneously. Together, these capabilities make the bioflux a powerful tool for designing multiplexed control studies that better reflect the human vascular environment. Information or want to request a demo, please reach out to us online. And with that, I thank you very much for your attention.
Thanks, Stephanie. That was a great and informational presentation. Before we move into the Q&A portion, we're ready to launch a short poll that will appear on your screen. If you could take a moment to answer this question as we move into the Q&A portion of the webinar. You can enter your questions using the Q&A box. And we already have a few questions that have been submitted, so let's jump in. The first question is, do all cells benefit from shear flow?
That is a great question. The cells that would benefit most from shear flow are those that in vivo conditions are also subjected to flow. So if we think about the cells that are in the vascular bed, in the circulature, those will absolutely benefit from shear flow because they are under constant flow in normal conditions.
Okay. Do I need to clean the bioflux after doing an experiment with blood or blood components?
No, you don't. So the beautiful thing about the bioflux is that from the electropneumatic pump to the plate, we're only moving air pressure. So the plates are really the only places that will ever contain blood or cells or any type of reagents. So all of the tubing will not need to be cleaned after an experiment because we're only moving air through it.
Yeah, I think our clients really appreciate that feature.
Yeah, it's great.
Which coatings can be used with the BioFlex plates?
Yeah, as we saw from the studies that were highlighted in the presentation, the plates can support various coatings. From the presentation, we saw that the channels were coated with collagen, with von Willebrand factor, P-selectin. They can also be coated with VCAM1, as well as with other ECMs that will support the establishment of an endothelial cell monolayer in the microfluidic channels.
Okay. And how many experiments do BioFlex plates support?
So that'll depend on the plate format that's used. As I mentioned during the presentation, we have plate formats that can support anywhere from 3 to 24, or that have 3 to 24 microfluidic flow cells that can run simultaneously. So if you choose the plate format that has 24 microfluidic cells, flow cells, you could run those simultaneously, or you could stagger them. There are many options, and there's a lot of versatility in how many flow cells you can run at a time.
Okay. Do you supply pre-coated plates, for example, coated with VWF or with collagen?
Yeah, even though that would be great, we do not at the moment, but we have coding protocols that we have developed with all of these ECMs or other coding factors that are very easy to do in the lab. So we don't have plates that are come pre-coded, but coding a BioFlex plate is very similar to how coatings are done in normal plates for static assays.
What are the sample requirements? What anticoagulant?
So that is a great question. That'll depend on the goals of the experiment. But generally, when blood is drawn, it'll go into some kind of anticoagulant, right? And then once that is loaded into the bioflux or into the bioflux plates, because we're working with microfluidics, we don't need large volumes of blood or any of the other blood components. We're really talking about microliter quantities of all of these fluids.
Um, Question of pulsatile versus laminar flow for these studies.
So the bioflux has the ability to include pulsatile flow to mimic the pulses that may be generated by the heart, for example. The bioflux has the ability to generate pulses in the flow to up to 2 hertz. So if we think about normal heart rate at 60 beats per minute, that would be about 1 hertz. So we can really accomplish anywhere from really low heart pulses to very high heart pulses when we're designing the experiments.
Okay. And which cell types have been used to create monolayers in BioFlex plates?
Right. So as we saw from the studies, HUVECs or human umbilical endothelial cells are commonly used for these studies. But from the literature, there's also new researchers have also used aortic endothelial cells. There are also papers showing that stem cells have been used to create monolayers in the bioflux, as well as epithelial cells. That's also another option.
Okay. And how does the system visualize platelet adherence, for example, if the sample is whole blood?
Right, so to do that, to differentiate between blood components, it is always a good idea to stain them in some way. So we could do antibody stains, or a very quick method to stain platelets is to use calcine AM, for example, although that will probably stain other things in the blood. But when we add these fluorophores or stains, then through imaging, we can parse out which blood components contributed to certain aspects of the hemodynamic processes.
Okay. Does your lab offer lab services with this shear flow system?
We actually do offer lab services. Yeah, so this would be we in our lab we have the versions of the bioflux that would support creating these lab services projects to test various things. Yes.
Okay. I think that's all the questions we have today. Thank you, everybody, for your engagement and for asking so many questions. As you leave the webinar, there will be a short survey. If you could answer those questions for us, we'd really appreciate it. We appreciate you spending part of your day with us, and we hope you